We're now ready to move into Chapter 17 in our microbiology class. We're continuing our discussions of the immune system. If you recall, in the last chapter, in Chapter 16, we were discussing the first part of the immune system that we called the innate immune system. Your innate immunity was made up by your first and second lines of defense. Your first line of defense were your skin and your mucous membranes. Your second line of defense were things like phagocytosis by your phagocytic cells, such as certain white blood cells, your neutrophils, and your macrophages. Other parts of your second line of defense were inflammation, fever, complement, all the different things we discussed in our last chapter. Now we're moving into the third line of defense, which is going to be made up of the immunity you have from specific cells in your body. As we go through all of this, keep in mind that for any of these things in your third line of defense to be activated, your first and second lines of defense have already been activated. So we've already had the pathogen get past your skin and mucous membranes. We've already had phagocytosis of your phagocytosis of the pathogen has occurred. Within your third line of defense, your adaptive immunity, you have two different branches of immunity. You have humoral immunity, which involves the B cell production of antibodies. This is called your humoral immunity because the antibodies are secreted into your bloodstream, and that is where they are going to become active. The other branch of your adaptive immunity is called cellular immunity, and this is going to be the response of your actual cells, specifically the function of your T cells. You have different types of T cells, which we're going to go over here in a few minutes, but the main important part of this cellular immunity is going to come from your helper T cells, which give permission, and then your cytotoxic T cells, which actually kill. Regardless of which type of cell we're talking about, both your T cells and B cells come from a similar origin. Inside of your bone marrow, you have specific stem cells. These stem cells have the ability to differentiate or change into multiple types of cells in your body. Some of these stem cells are going to migrate into your bone marrow, and throughout your life, they are going to be converted into B cells. That's where the B comes from, because they are grown and differentiated in your bone marrow. Other stem cells are going to move into your thymus, and then during your childhood, your thymus is going to lead the differentiation or the change of stem cells into your T cells. So that's where we get the T from. These cells are made in the thymus. As an adult, most of your stem cells have already differentiated into the T cells or B cells that they are destined to become. These T cells and B cells are now found in your body in many different places. Some are going to be in your blood, but they're mainly going to live in your lymphoid tissues, your spleen, your bone marrow, especially in your lymph nodes. Terminology changes slightly as we move into the adaptive immune system. If you recall from Chapter 16, we learned that the part of the pathogen attacked by the immune system cell was called a PAMP. Now we're going to more specifically talk about the parts of the pathogen that your immune system cells are going to respond to as antigens. Antigens can be anything that is exposed on the surface of a bacterial, protozoan, virus-infected cell, anything like that, that your antibodies can bind to, that your B cells can respond to, your T cells can respond to. This picture here is showing you a bacterial cell, and the reason you see several different shapes sticking off the surface is because the image is trying to demonstrate to you that one bacterial cell does not only have one antigen. There may be one protein sticking off of the bacterial cell surface that can be specifically recognized by this antibody. They're calling it antibody A. On the same bacterial cell, you'll have a different antigen sticking off that another antibody, antibody B, can bind to. The specific part of the antigen that the antibody or the cell binds to is called the epitope. Some of your adaptive immune system cells are going to respond to free antigen, meaning broken down pieces of the cell. Okay, so think about this. If I was a phagocytic cell and I ingested this bacterial cell, I would chew it up, break it into little pieces, and then spit the remaining pieces out. So what is on the surface of the intact bacterial cell will be some of the little pieces that get spit out. This would be a free antigen. Other cells are going to respond to the antigen on the bacterial cell, or possibly even something that is what we call presented by another cell. I want to show you a very quick video to demonstrate what I'm talking about right now. Recall from the last chapter that we understand this is a phagocytic cell. It's going to reach out and grab the pathogen and pull it in. We learned that already. But one thing we did not discuss was that this is not only called a phagocytic cell, it is also going to be called an antigen-presenting cell. Watch what happens. We're also now going to go a little deeper, and on this side we're showing you a human cell that's becoming infected with a virus. Both of these cell types, your human cell infected with the virus and the phagocytic cell, once they have the pathogen inside of them, the cell responds by taking a piece of the antigen, a piece of that pathogen, and putting it on the surface. It is presenting that antigen, that piece of the microbe. I like to think of it as the cell is kind of waving around a big red flag saying, hey, here it is, this is what I found. Once a cell starts displaying or showing this antigen to your immune system cells, we call this an antigen-presenting cell, or an APC. So we're going to see that as we go through this chapter. Since we started talking about antibodies and we see this antibody bound here to the bacterial cell, let's take a closer look at antibodies. The proper name for an antibody is an immunoglobulin. And some of you may have heard that term used, such as an immunoglobulin vaccine or injection, things like that. So we're going to learn what that means. If we take a look at this antibody or immunoglobulin over here on this side, you can see it kind of looks like a big Y. Every antibody in your body has this Y shape. It may be several Y shapes connected together, but they're all going to present this Y shape. The base or the bottom of the Y shape is made up of what we call the constant region. If something is constant, that means it never changes. Every antibody in your body is going to look exactly the same on this constant region. That's the way we know that they're your antibodies. Your immune system cells can recognize, hey, this is part of my antibodies because it always looks like this. The other end of our Y is what we call the variable region. It's called the variable region because that is the region that changes. It is the part of the antibody that is different depending upon which antigen this antibody is specific for. If this antibody was made against a specific antigen found on E. coli, the variable part of this antibody will bind to E. coli. It will not bind to anything else. It is only made against that antigen. Just to point out a couple other characteristics, notice we have what we call light chains and heavy chains. That simply means the light chain, which is in blue, it's smaller, so it weighs less. The heavy chain in green you see is larger, so it's heavier, it weighs more. Each of these separate chains are connected by what we call disulfide bonds. These are some of the strongest bonds in the world. They do not break once they are formed. There are 5 major classes of immunoglobulins or antibodies found in your body. If we use our same example, let's say I have an antibody that is specifically going to be able to bind to E. coli. I'm not just going to have one. I'm going to have 5 different classes of that antibody. The major type of antibodies you have are called IgGs. IgGs have the job of enhancing or helping phagocytosis. Their job is to simply bind to an antigen and kind of display, hey, here it is, I need a phagocyte to come eat this. This is not supposed to be in my body. IgMs are pentamers. They're 5 Ys stuck together. They're the big guys. They're the first ones that are going to initially respond to an invasion of a pathogen. If you think about it, that makes sense. If you've got an army and you're going to go fight somebody, well, you're going to send in the big guys first. And that's exactly what your body does. IgAs are found in all of your secretions and anywhere you have mucous membranes. They are found in extremely high concentrations in your eyes. Why would you need to make antibodies and specifically put them in your mucous membranes? Your mucous membranes are your first line of defense. That's going to be the first place that you may get an infection. You may have an exposure to a pathogen, so you need some protection there. IgDs, we're still a little uncertain on their function. IgEs specifically help eosinophils. And if you recall from Chapter 16, eosinophils are specifically made to remove parasitic worm infections from your body. So how do we get these antibodies? They're obviously important. That's what we've been talking about since we started this chapter. You get your antibodies from your B cells. They make up your humoral immunity. The way this is going to work, you are going to have B cells already made in your body. When the B cell recognizes an antigen, meaning it finds a pathogen that has been in your body, it has already received signals, cytokines, from the phagocytes, the second line of defense that it's already been activated. Your B cells are going to bind to the antigen and then undergo what we call clonal expansion or proliferation. We'll flip to a picture to show you what's going on. So here's these original stem cells in your body. They've already moved to the bone marrow and they've been made into a little army of B cells. Each B cell that is made responds to a different antigen. So now the particular antigen that has come into our body, they're showing it here in these little squares. This is a free antigen. How do we have free antigen floating around in our body? We've already had a phagocytic cell ingest the pathogen, break it down through the phagosome, fusion of the lysosome, into small pieces that are excreted out of the phagocytic cell. So in this particular example, this one B cell can specifically bind to this antigen. Notice the others can't, so they can't do anything. But the one that binds the antigen, then we are going to now call this our activated B cells. Activated B cells do 2 different things. They are going to expand and proliferate. Proliferate means they are going to multiply in number. Why are we multiplying this activated B cell? We need it, right? This is the B cell that recognizes the antigen. So this B cell can make antibodies that bind this antigen specifically. We now have built our little army of B cells by proliferating. The army of B cells mature into plasma cells. Plasma cells then secrete the antibodies that go into your bloodstream and bind any of these antigens that originally activated the B cell. Notice the antibodies are in the blood, and this is why we call this our humoral immunity. Making the plasma cells to produce the antibodies is not the only thing going on here, though, right? Some of our activated B cells produce what we call memory cells. These memory cells are not going to do anything right now. They kind of hang back and remember that they were once activated and that they may in the future need to respond to this antigen. 2 months later, 2 years later, 20 years later, whenever this antigen comes back in our body, we don't have to wait for this step to happen. Immediately, the memory cells can bind the antigen, turn into plasma cells, and make antibodies. We can get an antibody production much quicker during the second exposure because we made the memory cells the first time. We're going to talk about vaccines in just a minute, but let me go ahead and make the point while we're discussing it. This is the reason you get vaccinations. You need a memory in your body. So if you get a flu vaccine, they don't give you the whole flu, then nobody would want it. They gave you the flu. When they give you a flu vaccine, they don't give you the whole virus, just a little piece. That's all we needed, right, was just this piece of the bacteria. So your B cell will bind to the piece of the flu we gave you in the vaccine. It'll make immediate antibodies against the flu, but what's more important is that we make memory cells against the flu. So then later in the year, if you inhale that flu virus, you can have an immediate antibody production, an immediate immune response because you have those memory B cells against the flu. Now, unfortunately, it's not just quite that simple, but it's a good thing it's not quite that simple. Would it be bad if our B cells decided to bind to our liver cells? Yeah, then we'd be producing antibodies that could destroy our liver. We don't want to do that. So we have to have a regulation step. And that's what they're showing you here in this image. Here's our free antigens. One B cell can specifically bind to this antigen. Then we have clonal expansion, proliferation. What actually needs to happen before we can make the antibodies is this little guy has to come along, look at what the B cell has, and say, yep, you're right, you found something bad, and we need to get rid of it. So the TH, the helper T cell, actually binds to the antigen on the B cell and looks at it, and then sends a chemical signal to the B cell that says, yes, you may continue. If this B cell had accidentally became activated and was binding to one of your heart cells or your nerve cells, when the helper T cell looked at it, the helper T cell would say, no, you are not supposed to kill that. And that would prevent us from making antibodies against something we're not supposed to. Why are we making these antibodies? What do they actually do? The antibodies themselves are capable of 4 different activities. Antibodies can cause agglutination, opsonization, neutralization, or activation of complement. One thing they can do, called agglutination, is a fancy term for clumping. The antibodies will begin to bind all the different pathogens, making them into large clumps. The large clumps are easier for our body to get rid of. Opsonization is the fancy way of saying helping phagocytosis. Shown in this image, here's the phagocyte in red trying to eat the bacteria. If the bacteria is covered or opsonized by antibodies, it's easier for this phagocytic cell to ingest the bacteria. Neutralization means not that the antibody is going to kill the pathogen, but it keeps the pathogen from hurting us. Think about what we learned in Chapter 13 on viruses. Step 1 of viral infection in the lytic cycle was attachment. If the antibodies are surrounding the virus, shown here, then the virus can't attach to our cell. If it can't attach to our cell, it can't do anything. Same thing goes with a bacteria or a toxin. Other things the antibody can do is simply activate or kind of rush the other parts of the immune system, like complement. You should be beginning to see a trend that what we're doing is we have this giant army with all the different little parts, and we can't look at one part by itself. All the little parts help the others. Now let's take a look at what our T cells do. You have two major types of T cells, as we discussed earlier. You have helper T cells and cytotoxic T cells. Helper T cells, we've already seen, they regulate. We've already seen that they are the ones that give the B cell permission to make the antibody. The cytotoxic T cells become a cell that is capable of actually killing either a virus-infected cell or a bacteria itself. So let's move to the picture to see what happens. The T cells are a little different than the B cells in that they cannot respond to free antigen. A T cell requires presentation of an antigen. So recall from the video I showed you earlier, this can come from what's shown in the picture here, a phagocytic cell eating the bacteria, breaking it down into little pieces, and presenting or showing one of those pieces on its surface. If the phagocytic cell shows one of these pieces of antigen on its surface, the B cell, the TH, the helper T cell, will come along, look at it, and say, oh my gosh, yes, you're right, you have found something bad. And the T cell then starts sending out cytokines that are going to signal that we need to do something about this. Once the TH cell has realized that something is wrong, he can go tell B cells, make antibody, or he can go tell cytotoxic T cells, you need to start killing these bacteria. You can have a direct death of cell occur if a virus-infected cell presents an antigen to a cytotoxic T cell. The cytotoxic T cell will start sending out little perforins, little chemicals that make the infected cell just shrivel up and die. The cytotoxic T cell can cause this cell death in an entire bacteria, in one of your cells that's infected with a virus, in a protozoan cell, lots of different types of cells. So now let's look at an overview of what happened. Over here in this yellow-orange color, this is your humoral immunity. You have free antigen. This was made because a pathogen made it past the first line of defense, and then in the second line of defense, a phagocytic cell spit out little pieces of the broken down pathogen. The B cell binds to a free antigen, proliferates, makes a plasma cell to produce antibodies. These antibodies can go back and bind to the original antigen. Remember, to do this, the helper T cell has to give the B cell permission. At the same time, the phagocytic cell in the second line of defense will present some of this antigen to your helper T cells. The helper T cells send out signals, of course go tell the B cells what to do, but also give permission to the cytotoxic T cells. Cytotoxic T cells say, all right, got my permission, you showed me what to kill, and then they start killing cells. In addition to the ability to produce antibodies and activated cytotoxic T lymphocytes to kill an immediate threat, some B cells that are activated make memory cells. The memory cells will respond the next time we get a pathogen invading our body. They will become plasma cells to produce antibodies. Now that we've finished discussing the immune system, we can talk about types of immunity you can receive. You can have natural immunity or artificial immunity. Natural immunity means that you were exposed to an antigen naturally. Artificial immunity means that you were exposed to an antigen artificially. Natural exposure would be something like I sneezed on you, you kissed me, you drank out of the same glass I drank out of. All those are natural ways of transmitting a disease. Artificial immunity would come from, say, an injection of some sort. That's not natural. That injection was man-made. That needle was made by man and then stuck into your arm. You wanted that exposure to happen. It didn't just naturally happen. Immunity is then classified as either active or passive. Active immunity requires your body to be exposed to the antigen and then respond by both your T cell being activated and your B cell being activated to make antibodies. All those things we just went over. If it's active immunity, your body has to do all of those things. If it's passive immunity, that means you are getting the antibody already made. So your T cells and B cells don't actually have to do anything themselves. So now let's put those together. A natural active immunity is going to be when the person is exposed to an antigen naturally. They get sick and then recover. What has happened is the antigen gets into the body. The body then makes antibodies and cytotoxic T cells are activated. That's why you get better. You kill the pathogen. But the key here is that you make memory cells, so you now have an ability to fight it the next time it comes in your body. Natural passive immunity is when by a natural means antibodies are directly given to you that can fight an infection. An example of this would be breastfeeding. When a mother breastfeeds her child, every antibody that is in the mother's blood is transferred into the mother's milk in the form of colostrum. So the mother gives her antibodies directly to the baby. The baby's B cells didn't have to do anything themselves. They just get the antibody. Artificial active immunity is what we call a vaccination. Whenever you are vaccinated against the flu or tetanus or anything like that, we don't actually give you the entire bacteria or the entire virus. That would make you sick. We give you just a small enough piece of the pathogen that it can't make you sick, but a big enough piece that your B cells and your T cells will respond to it. So if we talk about the tetanus shot, we give you just a little piece of a cell surface protein of the bacteria that causes tetanus. Your B cells then make antibodies and memory cells against that bacteria. So if you get the bacteria in your body, you do not get a disease. Your immune system is ready for it, and it can immediately kill that bacteria when it comes into your body. Why do we need booster shots? Your body can only hold so many memory cells. If your memory cells are never challenged, meaning your memory cells never see tetanus, then they will eventually degrade and they won't be good anymore. So every so often we have to get booster shots so that our body can remember what that bacteria or that virus looks like. Artificial passive immunity is very rare. This is going to be an artificial introduction of an antibody into your body. This would be something like an immunoglobulin shot. If I have immunity against different things, and let's say my daughter is diagnosed with some devastating leukemia or some type of cancer of the blood that makes her body no longer able to make antibodies, then they can take some antibodies from my body, put it into a shot, and then give that shot to my child. That would be giving her the antibodies, giving her the immunity. Her body's not having to make the antibodies, but she's now going to be partially protected from any sort of invasion. That concludes our discussion of the immune system. I highly recommend that you really sit down and think about this in the kind of the sense that we've been going through it. Your immune system is an amazing, highly organized army that is sitting there waiting for the introduction of pathogens into your body. And that army is going to fight that pathogen from every different direction.